Thank you, Doug. Certainly, as the committee approached, one thing that I felt like vastly needed in the committee as well is trying to really include ileal amino acid digestibility of feed ingredients. So that's really key in terms of formulating diets accurately. And we know that we heard from Dr. Kidd this morning and talked about the committee that met in 2004 to really establish methodology, and from that, we've had some published papers in the literature on some really key ingredients, and so we certainly wanted to set out to really look at some of these key papers and come up with summarizing some of this data. So, research methodology, we can all attest to this, has been a really key situation going through a lot of these papers. You really may ask, well, why didn't the committee use this paper or that paper? And, we set out setting criteria so that, really we had some, scientific integrity going through this, because the NASEM committee, they really expect a high level of research integrity. So the committee really set some criteria, and this is what we really looked at in terms of our, ingredient papers out there in terms of amino acid digestibility. We really set 1992, because certainly on some of our ingredients, as we all know, certainly like corn and soybean meal, certainly yield has a significant impact on amino acid composition, for example, also as well as others. So we do see this with a lot of this. And when we take a look at distillers grains, before 94, we really, it was a totally different ingredient that we see today. I mean, since 2004, certainly ethanol production has increased. So this is an ingredient that's really been impacted here recently over the past several years. So really tried to look at this published data and tried to look at adequate methodology in terms of detail. Really had a strong preference on ingredients that would be commonly fed to poultry species, particularly in the U.S., and looked at ingredients, really macro ingredients with really 5% or higher. And we looked at about 22 journals for the initial selection of amino acid digestibility, really to define these coefficients. So this did take a lot of work going through some of this. So from that, the new report will have amino acid digestibility of about 14 different ingredients. So just to give you an idea. Kurt talked about new content, and particularly on, for graduate students, we really had that in mind on, really expanding this publication. I believe he put up there's 1,050 pages in this publication. So with the amino acid chapter, certainly we wanted to do some expansion there. So in terms of bioavailability and analysis, I have it in 3 categories. I've got requirements, and then you'll see deficiencies and relationships. So, really talk about sources of amino acids, whether they're intact or either synthetic or crystalline. What is the bioavailability of D amino acids? There are certain amino acids that certainly have some availability, where there's others that have virtually zero availability. So that was an area that I really feel like can be beneficial to our students on. And then we included amino acid analysis of feedstuffs as well as physiological fluids. So there's a big difference there of how we approach the analysis on feedstuffs versus fluids. In terms of requirements on empirical estimates, really what's true and dear to my heart is how can we teach people to conduct an empirical study correctly? And a laying out what is really essential and what we need. So really spent some time going through that to be able to teach people what we really need to really identify a really good, well-designed experiment. And then what are some of the factors that influencing variations in requirements, and what is some of the dogma? And certainly for an example, we tend to think with temperature, birds eat less, we can feed more amino acids, but then when we get into heat stress conditions and it starts impacting something like T3 and T4, and that impacts protein accretion, then that certainly can impact our requirements. So that's just an example there. But did go through several factors impacting requirements, and then deficiencies and relationships, and I really see this being very important moving forward. Amino acid deficiencies, this can be really useful for the academic side in terms of teaching amino acid deficiencies in basic courses. Then we've got a section on imbalance, antagonism, and toxicity. I then went into expanding the role on the previous edition on specific amino acid relationships. So, really talk about cysteine and cysteine and really want people to understand that the difference there, as well as how that relates back to methionine through metabolism. That was a section that we really wanted to expand so that graduate students could have an appreciation. One that's come up here recently has been glycine plus serine, and some of us know glycine plus serine. Glycine's a semi-essential amino acid, particularly in the starter period. In terms of amino acid requirements, we really looked at factorial modeling as well as empirical methodology. And then in terms of methodology, I think what's really important is having treatments range from about 70 to 130% to really optimize that plateau so that we can really achieve an appropriate slope to have adequate requirements. Having enough spread in these amino acids is really key from a 0.05 to a 0.10 so that we can, and then really have an appropriate level of biological replication. Some times 8 replications is enough, but as we get into these heavy birds around 3 to 4 kilos, we may need about 12. So then in terms of amino acid requirement assays, I think it's very important to analyze intact amino acid sources for composition and report those, as well as digestibility coefficients that were used in a paper. Is encouraged to obtain digestibility values for our treatments during experimentation. Also, look at if we have an uncommon ingredient used to create a deficient situation, really characterize that ingredient very well. And then use a requirement assay to be at least 7 days. The reason for that, the blood turnover, it takes about 7 days for an amino acid to turn over. Maximum on 21 days as these birds grow extremely rapidly. So, statistical, we know that we can use linear and non-linear models and really look at some of these confidence intervals, AIC, and standard error, the estimate, I think is very important. And so, these requirements can be very time consuming and certainly very expensive, so we've got to look at some other applications. I know that looking at digestible ratios is very important, but again, I think long term we're probably going to have to look at factorial modeling. Just an example of glycine specific relationship being semi-essential. Here's an example I talked about earlier about temperature on T3. Certainly high temperatures can really affect protein accretion in the birds. So in summary, accurate amino acid digestibility coefficients for feed ingredients are needed in formulation. These What we encountered was digestibility coefficients are limited for peanut meal, corn co-products, poultry byproduct meal, and fibrous ingredients used in broiler breeder and pullet formulations. So, it's important to provide as much information about an ingredient source when you're publishing amino acid digestibility work. Empirical amino acid digestibility, just design these trials correctly, put a lot of time in that, and we do realize and acknowledge it's very laborious. There's a lot of cost associated with implementing these empirical assays. So, I really think as an academic side, we've really got to start looking at modeling for the future, and just last, this amino acid chapter was expanded on previous editions, and this kind of concludes this presentation.